Hello and welcome to WGN, Whole Genome Network. On today's segment, we will be learning about whole genome sequencing. I'm your host, Liam Byrne, and today we have two guest scientists joining us, Elena Van Ziska and Madeline Wong. So, what is whole genome sequencing?
Whole genome sequencing is analyzing the genes in the entire nucleotide sequence of an organism. It is a report of the decoded nucleotide bases, seeing the order in which the letters adenine, guanine, thymine, and cytosine come in.
If wanted, you can discover the sequencing behind one gene only. However, whole genome sequencing looks at all the genes of a specific genome and its letters.
Did you know that the whole human genome has nearly 3 billion nucleotides? High-tech machines are used to decipher the nucleotide code after scientists have broken down the genome into smaller segments. Interpreting and analyzing takes the longest, as the letters, though in an order, may make no sense at all. Scientists start with no idea of what the letters are assembled to produce.
Imagine a paragraph from science or English with enormous words that just don't make sense. In order to make sense of these words, you have to do some research and risk a little trial and error. Sequencing of genetics is often like putting together an enormous biological jigsaw puzzle.
Nowadays, we have advanced technology that allows us to read large segments of DNA quickly.
This is called next-generation sequencing. It replaced the previous Sanger sequencing, which was named after the scientist Frederick Sanger. Though effective, the process of using Sanger sequencing is extremely expensive and does take a really long time.
With next-generation sequencing, the human genome can be deciphered within a few days for under $10,000, whereas through Sanger sequencing, the human genome took 13 years to decipher and $2.7 billion.
Sequencing of entire genomes are instrumental to health sciences. It is used in identifying and diagnosing inherited diseases and disorders, characterizing mutations, tracking disease outbreaks, and learning more about cancer progression. Majority of diseases they look into are sourced by genetic mutation. Hence, looking at the entire sequence will identify the base and what steps should be taken to treat the illness.
Once a report is given, scientists and doctors can analyze particular genes at a much quicker rate. Besides using it to diagnose diseases and design custom treatments, genome sequencing is used to identify mutations that will lead to illnesses in the future before symptoms appear. By interpreting information of someone's potential risk of genetically caused diseases, doctors could begin to formulate a personalized plan to intervene early and improve their quality of life.
During sequencing, it captures a base-by-base view of the genome in high resolution, revealing large and small variants that may have been missed. Genes account for only a quarter of DNA in the genome. With an entire sequencing decoded, scientists can do further research on the unknown. So let's review what we've learned.
Whole genome sequencing is decoding the order of the nucleotides in all the genes of a specific organism, such as humans.
Interpreting and analyzing the DNA code is always difficult at first.
We discovered the sequences through a cheaper and faster way, now called next-generation sequencing.
It replaced the Sanger sequencing process, which takes an extremely long time and a very large amount of money.
Looking at the whole genome sequencing of people is helpful, especially in healthcare. Doctors are able to identify and diagnose inherited diseases, spot mutations before symptoms appear, and formulate personalized treatment plans to suit the patient best.
And that, viewers, will wrap up today's episode on whole genome sequencing. Thank you for joining the Whole Genome Network, and we hope to see you next time.